Welcome to the learning module for Group 2 hormone signaling mechanisms for G-protein coupled receptors and second messengers. This is Dr. Tefke for the Cellular and Molecular Medicines course. After studying this learning module, students should be able to describe the structure of G-protein coupled receptors, explain how heterotrimeric G proteins function to transduce signaling from G-protein coupled receptors, describe how the activity of the alpha subunit is regulated by GDP and GTP, explain the differences between the three main classes of G protein alpha subunits and which effector enzymes they modulate, explain how cyclic AMP activates protein kinase A, describe the second messengers produced by phospholipase C, explain how intracellular calcium functions as a second messenger and the role of calmodulin in transducing calcium signaling, describe how cholera toxin and pertussis toxin affect G-protein coupled receptor signaling pathways, and explain how defects in G-protein coupled receptor signaling pathways contribute to human diseases. G-protein coupled receptors are a large family of cell surface receptors that all function by a very similar mechanism. When bound by a ligand, they interact with a group of proteins collectively called heterotrimeric G proteins. The G proteins then become activated and stimulate or modulate the activity of another enzyme called an effector enzyme, or open and close ion channels in the membranes of cells. Activation of effector enzymes alters the concentration of second messengers within the cell that ultimately controls intracellular signaling pathways to produce a cellular response. In general, G-protein coupled receptors bind to small peptides of 8 to 12 amino acids in size, as well as other very small molecules such as amino acids or nucleotides. All members of the G-protein coupled receptor class share a common structural feature that includes 7 transmembrane spanning domains or alpha helices. The N-terminal domain is extracellular and along with 3 extracellular loops contributes to binding of ligands or hormones. A C-terminal domain on the cytoplasmic side has an interaction domain which can make contact with heterotrimeric G protein subunits. The presence of 7 transmembrane spanning domains is a defining characteristic of all G protein coupled receptors. Let's begin by looking at the general mechanism for activation of the effector enzyme adenylate cyclase by a hormone or neurotransmitter interacting with the G-protein coupled receptor. We begin in the unstimulated state in which the receptor is not occupied by the hormone or neurotransmitter. The heterotrimeric G protein has 3 subunits, alpha, beta, and gamma. The alpha subunit contains a guanine nucleotide binding domain. In its inactive form, the alpha subunit is bound by GDP. Hormone binding to the G-protein coupled receptor causes a conformational change that allows the C-terminal domain of the G-protein coupled receptor to contact the heterotrimeric G protein. This causes a conformational change in the alpha subunit of the heterotrimeric G protein, such that GDP is released and GTP binds to the alpha subunit guanine nucleotide binding pocket. This activates the alpha subunit, causing it to dissociate from the beta and gamma subunits. The alpha subunit with GTP bound to it can then make contact with effector enzymes in the plasma membrane. In this case, the effector enzyme is adenylate cyclase, the enzyme that converts ATP to cyclic AMP. This increases the intracellular concentrations of cyclic AMP, the second messenger in this system. The alpha subunit has an intrinsic enzymatic activity called a GTPase. A GTPase will hydrolyze the gamma phosphate of GTP, converting it to GDP. Now the alpha subunit has GDP in its binding pocket. This switches it off by causing a conformational change that renders it unable to make contact with adenylate cyclase to stimulate its activity. The alpha subunit with GDP can then reassociate with the beta-gamma subunits and form the heterotrimeric basal state G-protein complex until stimulated again by another G-protein coupled receptor. There are approximately 20 different subclasses of heterotrimeric G-proteins, primarily differing in the alpha subunit's ability to contact a variety of effector molecules or enzymes. The three primary alpha subunit families are G-alpha S, which stimulates the activity of adenylate cyclase to raise intracellular cyclic AMP levels. The G-alpha I subfamily inhibits adenylate cyclase and reduces intracellular cyclic AMP levels. And the G sub Q class of alpha subunits, which are also known as the G sub P or G sub PLC family, since they activate phospholipase C to generate two second messengers, IP3 and diacylglycerol. In all cases, it is still the alpha subunit that contains the guanine nucleotide binding domain. The GTP-bound form is the active form, and the GDP-bound form is the inactive form. The overall activity of adenylate cyclase in a cell is the net effect of cell signals acting through G-alpha S coupled G-protein coupled receptors and receptors that are coupled to G sub I. This allows fine-tuning of the cyclic AMP levels in the cell to a complex array of extracellular conditions. Cyclic AMP works well as a second messenger because it is very rapidly generated from ATP by the activity of adenylate cyclase and also very rapidly degraded by an enzyme known as cyclic AMP phosphodiesterase. The ability to rapidly synthesize and rapidly degrade cyclic AMP means that signal transduction mechanisms functioning through cyclic AMP can be quickly turned on and quickly turned off. Methylxanthines, such as caffeine or theophylline, inhibit the activity of cyclic AMP phosphodiesterase and therefore prolong the effects of neurotransmitters and hormones functioning through G-protein coupled receptors that stimulate adenylate cyclase. The ability of methylxanthines to inhibit cyclic AMP phosphodiesterase only partly explains their stimulatory effect. The primary stimulatory effects of caffeine and theophylline are due to antagonism of adenosine receptors in the central nervous system. Increasing the cytosolic concentrations of cyclic AMP leads to activation of a serine threonine kinase known as protein kinase A, or pKa, or sometimes cyclic AMP dependent protein kinase A. For the purposes of cellular molecular medicine, the molecule is called protein kinase A, or pKa. Protein kinase A is a heterotetramer with two regulatory subunits and two catalytic subunits. In its inactive form, the regulatory subunits are bound to the catalytic subunits and mask their kinase activities. The regulatory subunits have binding pockets for cyclic AMP. When cyclic AMP binds to the regulatory subunits, they dissociate from the catalytic subunits. The catalytic subunits then are activated as kinases and phosphorylate substrate proteins, affecting their activities, which produces the cellular response. Blood glucose levels are modulated by hormones. Glucagon and epinephrine bind to G-protein coupled receptors expressed in liver and in muscle tissue. When they bind to their receptors, they stimulate the production of cyclic AMP by activating adenylate cyclase. Cyclic AMP accumulates and binds to the regulatory subunits of protein kinase A. The catalytic subunits then dissociate and become active. Protein kinase A can then phosphorylate enzymes involved in both glycogen synthesis and glycogen breakdown. One of the substrates for protein kinase A is glycogen synthase, which it phosphorylates and converts to an inactive enzyme. This stops the synthesis of glycogen from glucose. Another substrate for activated protein kinase A is phosphorylase kinase. pKa phosphorylation of phosphorylase kinase converts it to its active form. Phosphorylase kinase can then phosphorylate glycogen phosphorylase and activate it. Once activated, glycogen phosphorylase breaks down glycogen to glucose for release into the bloodstream. Hormones that signal through G protein-coupled receptors to increase the level of cyclic AMP within cells can also affect the expression of specific genes in the cell. Activated protein kinase A can phosphorylate cyclic AMP regulatory element binding protein, or CREB. CREB is a transcription factor that binds to cyclic AMP response elements located in the promoters of genes sensitive to cyclic AMP regulation. Thus, short-term changes in cyclic AMP levels within the cell can be translated into long-term changes in gene expression. Signaling molecules that activate G protein-coupled receptors that are coupled to the G sub Q family of alpha subunits activate the effector enzyme phospholipase C. Phospholipase C cleaves phosphoinositide bisphosphates, or PIP2, located on the inner leaflet of the plasma membrane. This produces two second messengers, inositol trisphosphate, or IP3, and diacylglycerol. IP3 freely diffuses through the cytosol and binds to IP3-gated calcium release channels located on calcium storage vesicles such as the sarcoplasmic reticulum or the endoplasmic reticulum. As a result, intracellular levels of calcium rise and calcium-dependent processes become activated. Diacylglycerol, along with calcium, bind to and activate another serine-threonine kinase called protein kinase C. Protein kinase C is analogous to protein kinase A in that it will phosphorylate substrate proteins and alter their activities. Many calcium-dependent processes are controlled by a multipurpose intracellular calcium receptor known as calmodulin. In the absence of calcium, calmodulin is an alpha helical rod with a hinge region in the center. On both ends, it has calcium binding domains. Binding of calcium to calmodulin causes calmodulin to fold along this hinge region and clamp down on proteins that are regulated by calcium and calmodulin. A primary target of calcium calmodulin regulation is a serine-threonine kinase known as calcium calmodulin dependent kinase 2, or CAM kinase 2 for short. CAM kinase 2, in the absence of calcium and calmodulin, is inactive. It has an inhibitory domain which masks its catalytic kinase domain and prevents it from being active. When calcium levels rise, calmodulin clamps onto the inhibitory domain of CAM kinase 2. This allows the catalytic domain to become active. The catalytic domain then phosphorylates a serine residue in the inhibitory domain of CAM kinase 2. CAM kinase is now fully active. When calcium levels fall, calmodulin dissociates from the activated CAM kinase 2. However, the CAM kinase 2 remains active as long as the serine is phosphorylated in the inhibitory domain. Eventually, protein phosphatases reverse the phosphorylation and CAM kinase 2 reverts back to its inactive form. Here's a brief summary of G-protein coupled receptor signal transduction mechanisms. First, we have G-protein coupled receptors that couple to alpha sub S of the heterotrimeric G-proteins. G-protein coupled receptors coupled to alpha sub S heterotrimeric G-proteins stimulate adenylate cyclase activity and increase cyclic AMP levels, which activates protein kinase A. G-protein coupled receptors, which are coupled to alpha sub I, will inhibit adenylate cyclase and reduce cyclic AMP levels in the cell, thus reducing the activity of protein kinase A. Receptors that are coupled to G sub I alpha subunit heterotrimeric G proteins will activate phospholipase C. Phospholipase C cleaves PIP2 in the plasma membrane to produce two second messengers, inositol trisphosphate or IP3 and diacylglycerol. IP3 diffuses in the cytosol to bind to IP3 gated calcium channels on calcium storage vesicles and inducing the release of calcium into the cytosol. Calcium and diacylglycerol together fully activate protein kinase C, which will phosphorylate proteins to induce cellular response. Many of the calcium-dependent processes within the cell are mediated by calmodulin, a calcium-sensing protein in the cytosol. When calcium activates calmodulin, it interacts with other proteins and controls their activity, such as calcium calmodulin-dependent kinase 2. Let's look at some examples that demonstrate the clinical relevance of G-protein coupled receptor signaling. Cholera is caused by Vibrio cholera colonizing the intestinal tract and secreting a toxin known as cholera toxin. Cholera toxin acts as an enzyme which ADP ribosylates the G-alpha S subunit. The ADP ribosylation inactivates the GTPase activity of the alpha S subunit. This means that the alpha S subunit remains locked in the GTP bound form, leading to a persistent pKa-mediated phosphorylation of proteins, including the cystic fibrosis transmembrane conductance regulator, or CFTR protein, which controls chloride and water transport across the intestinal epithelium. Protein kinase A stimulated CFTR activity then produces a life-threatening diarrhea. A similar situation causes whooping cough, which results when Bordetella pertussis colonizes the respiratory tract, while cholera toxin ADP ribosylates G-alpha S subunits and inactivates their GTPase activity, thus promoting excessive adenylate cyclase activity. Bordetella pertussin produces pertussis toxin, which, like cholera toxin, functions as an ADP ribosylase enzyme. Pertussis toxin ADP ribosylates selectively GI alpha subunits. This results in inactivation of the GTP-GDP exchange mechanism, such that GDP-GTP exchange cannot be stimulated by hormone signaling. Thus, signals that would normally result in inhibition of adenylate cyclase are ineffective. The net result is unchecked adenylate cyclase activity in the cells, resulting in the production of cyclic AMP and activation of protein kinase A, which interferes with cellular signaling mechanisms controlling phagocytic clearance of infections. Since the treatment of cholera and whooping cough does not involve altering ADP ribosylation of alpha subunits of heterotrimeric G proteins, this may seem like extraneous detail to learn. However, questions about the mechanisms of cholera toxin and pertussis toxin have been a mainstay of the Step 1 and shelf exams for as long as I've been teaching this material. Parathyroid hormone regulates serum calcium and phosphate levels. If blood calcium levels are too low, the parathyroid glands secrete parathyroid hormone, which causes calcium to be released from the bone and the kidneys to reduce the secretion of calcium into the urine. Pseudo-hypoparathyroidism or Albright's hereditary osteodystrophy, is a rare genetic disorder that resembles hypoparathyroidism. Patients with pseudo-hypoparathyroidism have low serum calcium and high phosphate levels, yet their parathyroid hormone levels are appropriately elevated. Persons with AHO display a constellation of clinical features including short stature, obesity, round faces, subcutaneous ossifications, and shortened metacarpals associated with hypocalcemia and hyperphosphatemia. It is caused by a lack of response to parathyroid hormone, rather than a deficiency in parathyroid hormone. This is an analogous mechanism to generalized thyroid hormone resistance and androgen resistance discussed in the nuclear hormone receptor learning modules, except that the parathyroid hormone receptor functions correctly. The defect is due to a mutation in the GNAS1 gene that encodes the alpha S subunit. When bound by parathyroid hormone, the parathyroid hormone receptor cannot sufficiently activate adenylate cyclase. Persons with pseudo-hypoparathyroidism also display a lack of responsiveness to other hormones signaling through G-protein coupled receptors coupled to G-alpha S. Growth hormone, also known as somatotropin, is a peptide hormone that stimulates growth and cell reproduction. Growth hormone is secreted from the pituitary gland in response to growth hormone releasing hormone receptor activation, which is a GS-coupled receptor. Spontaneous mutations in the GNAS1 gene affecting the GTPase activity of the alpha S subunit can occur in pituitary somatotroph tumors. The result is hyperactive adenylate cyclase activity, which produces excess cyclic AMP and thus excess protein kinase A activity. These cells thus continually secrete growth hormone. Prolonged growth hormone excess thickens the bones of the jaw, fingers, and toes, resulting in heaviness of the jaw and increased size of the digits. This collection of features is referred to as acromegalia. If it occurs in early childhood, pituitary gigantism is the result. Some relatively famous individuals have suffered from acromegalia and pituitary gigantism, including Andre the Giant, Ted Cassidy, or Lurch from the Addams Family, and Richard Creel, probably most famous as Jaws in the James Bond movies. Similar mutations in pituitary corticotroph tumor cells results in oversecretion of ACTH, which stimulates the adrenal glands to produce glucocorticoids and cortisol. This is known as Cushing's disease. Here are a few questions to test your mastery of G-protein coupled receptor signaling pathways.